This protocol makes it possible to guarantee a good isolation of extracellular vesicles and identification of a higher number of EV-derived proteins. Compared to other isolation techniques, this one maintains the EV's morphology, integrity, and biological activities. EVs are being studied more and more in healthy as well as pathological conditions. This method can be applied to any system that wishes to characterize and study the EVs for their content or their biological effects. Special attention is required when isolating EVs, as this will affect their amount and the quality of the results in the following experiments. A visual demonstration is critical to show specific equipment such as a homemade size exclusion chromatographic column, as well as the involvement of nanoparticle counter and mass spectrometer.
Helping to demonstrate the procedure will be Antonella Raffo-Romero, a postdoc from the laboratory.
Start by pre-isolating extracellular vesicles, or EVs, from conditioned medium. Transfer the conditioned culture medium from microglia or macrophage cultures into a conical tube, and centrifuge it at 1,200 times G for 10 minutes to pellet the cells. Transfer the supernatant into a new conical tube, and centrifuge for another 20 minutes to eliminate apoptotic bodies. Then transfer the supernatant into a 10.4 milliliter polycarbonate tube. Place the tube into a 70.1 Ti rotor and ultracentrifuge it at 100,000 times G for 90 minutes at 4 degrees Celsius to pellet the EVs. After centrifugation, discard the supernatant and resuspend the pellet in 200 microliters of 0.2 micrometer filtered PBS. To isolate the EVs, prepare a